Hello everyone, this is Dr S S Bornare, Department of Genetics and Plant Breeding, KKV College of Agriculture, Nasik. So friends, today we are going to learn our next practical exercise of GPB 355, that is emasculation and hybridization in sorghum and pearl millet. So let us see first the emasculation and hybridization technique in the sorghum. Before we going to detail in this is the just taxonomy or the very floral biology of the sorghum. The common name of sorghum is jawar. Then scientific name is sorghum bicolor. Then chromosome number is equal to 20. Origin is in Ethiopia, and these are the two progenitor. That is sorghum arundinacea and sorghum sudanese. These are the two progenitor species of the sorghum bicolor, which is normal our sorghum. The flower structure of sorghum, these inflorescence of sorghum varies from the compact head to the open panicle. So there are two types of flower inflorescence in the sorghum. One is compact and another is open. The spikelet always occur in a pair. One is sessile and other is pedicellate, except the terminal, in which in terminal, rather than this terminal spikelet, the all other spikelet are always in the pair and having one sessile. That means the without pedicel or stalk of the flower, and one flower is pedicellate. Then flowers are also called as a floret. Due to the tiny flower in sorghum, it is called as florets, and they are enclosed by two bracts called as pelia and lemma. So as we see in all cereals and millets, there are the outer bracts called as lemma and pelia, which is the enclosing hull of the cereal. The lemma encloses the pelia of the floral organ. The florets occur in the group called as spikelet, and each spikelet subtended by bracts called as glumes. The perianth type, this perianth means when the calyx and corolla are not differentiated, they are fused together. They are called as special type of structure called as perianth. And in the all cereals, mostly these calyx and corolla are not available. Instead of that, there are a modified form of the calyx and corolla, that is perianth, is present. And perianth of this each floret is represented by two transparent scale called as laudicles at the base of the ovary. There are generally three stamens and gynoeciums with the two hairy stigmatic lobes. The stigmatic trichomes are designed to capture the wind-borne pollen. We can see here. This is the overall overview of the floral biology of the sorghum, in which we can see the panicle or the spike of the sorghum, which is having central rachis or rachilla and having the number of spikelets, that is group of florets, are arranged over them. This is the spikelet. Each spikelet consists of pairs of floret. One is sessile, that means it does not have the pedicel, and another is a pedicellate one. You can see here. And this each floret having the outer glume and the called as lemma and pelia, and in between this lemma and pelia there are three anthers and the bifid or bifidaries stigma, and at the base of that ovary there are laudicles are present. You can see here, these are the laudicles. So this is the overall floral biology of or flower structure of the sorghum. Then what is floral biology? That means what are the timing of anthesis and stigma receptivity. Let us see here the floral biology. The sorghum inflorescence bloom in the early morning. The anther and stigma push out and as the glooms open, and stigma are receptive for 1 to 2 days before the blooming, up to the 18 to 16 days after blooming, depending upon the variety and climatic conditions. And stigma before the antheridiaceans are subjected to the cross pollination. And this sorghum is a typical example of the often cross pollinated crop. Due to the presence of cross pollination leads to the 5 to 50%. So that's why this is the case of often cross pollinated crop. And in this sorghum, the stigma receptivities don't have any problem of stigma receptivity. As we can see here, the stigma is receptive from entire length of the flowering or blooming, and the cross pollination is greater in variety with open panicle than in the 14 compact panicle varieties. So this is the comparison between the open and compact panicle types of variety. Anthesis, that is the anthrodehiscence or first opening of the flower, are reported or begin from 2 a.m. and opening continues till 8 a.m. So this anthesis timing is early in the morning, very early in the morning, may prolong to the 10 a.m. And fertilization takes place 1 to 2 hours after the pollination. So this is the all floral biology of the sorghum. We have to take care for the efficient hybridization. Then what is the hybridization technique? So in case of hybridization technique, first is the emasculation. As we all know that emasculation means the removal of the male plant part, that is anther, from the female before the pollination, so that the female lines are having only the female part, and most of the cereals are bisexual. That's why we have to compulsorily emasculate the male plant part from the female line, and thus we can use the female in the hybridization and designated as a one male parent and another is female parent. And then this emasculation can be done by using hand emasculation or emasculation by hot water treatment, or used by the male sterile line or the plastic bag or mass emasculation techniques. So these are the different techniques through which we can achieve the emasculation. But in case of the sorghum, we mostly prefer the hand emasculation, and for that we have to choose the panicle or emerging panicle as given in the pictures. At the time of this this panicle is emerging from the flag leaf, we can see here. Then after selection, remove the upper part which is likely to be mature, and also the lower and immature part, and select the in between of the panicle, where the maximum chance of that the anthers are immature, and by using the forceps and remove the anther from the each floret. So this is the tedious one job. We can see here the we have to remove the anthers from the each of these spikelets, and this is the emasculation. We can see in the diagram also here the opening of the each floret and removing the anthers. Here by opening this, remove the 3 anthers, and after emasculation in the female flower, next day morning there is a pollination, that is hybridization technique. These pollinations, the pollens are collected in paper for few hours after the dehiscence from the male parent, and collected pollens are uniformly spread on the emasculated panicle of the female parent. So this is the crossing between the emasculated female line cross with the pollen from the male parent. So this the progeny is derived is F1, which is the hybrid seed. So we have to emasculate the female flower and bag in the male flower, and in the morning we open this bag and collect the pollens, and these pollens are dusted over this female parent. So this is the hybridization technique, and the fertilization takes place 2 hours after the pollination, and after that the fertilization takes place. That means the fertilization means the germination of pollen grains, and then pollen tube is formed, and the male gamete is fused with the uh, embryo sac of the female. The development of embryo and endosperm continues along with the starch accumulation for about 30 days, and after 30 days we can harvest the hybrid seed from that F1 hybrid seed from the female parent. So this is the hybridization technique in sorghum to produce the hybrid seed. You can see here the pollen grains of the male parent is bagged and pollen grains are saved, and in the next day morning we collect the pollen grains from that particular male and dusted over the emasculated female here. We can see here, and the pollens from the particular designated male and the ovaries of the or the female part of the designated female parent and cross between these two leads to production of the F1 hybrid. So this is the case of sorghum. Next is the pearl millet, or also called as the bajra. Botanical name is the Penicitum glaucum. Then family is Gramini. Origin is Western Africa. And distribution, the pearl millet is cultivated mostly in the Africa and in Asia also. The area is given here. The chromosome number of that particular pearl millet is 2n is equal to 14. So this is all about the taxonomy or introduction of pearl millet. Then floral biology. This pearl millet or bajra is a naturally cross-pollinated crop or allogamous crop due to the condition called as protogyny. This protogyny means gynoecium mature before the androecium, and this leads to the cross-pollination in particular crop, that is bajra. And inflorescence consists of the central rachis covered with short soft hairs and bear the fascicle on the rachilla. We can see the what is meant by the fascicle in the next slide. The density of the fascicle and length of the rachilla determine the degree of compaction of the panicle, and this each fascicle, let us see here, fascicle, this is the fascicle. Here we can see also, this is the fascicle. This is the fascicle. Each of these is called as fascicle. This is all inflorescence having central rachis on, and that particular rachis here, each and every the structure are having the bristles, and all the female parts are called as fascicles, and these are attached to the central rachis of that particular spikelets. So the density of these fascicles and length of the rachilla is determined degree of compaction of the panicle, and each fascicle consists of spikelet surrounded by walls of the bristle, that is involucre, and the prolongation of fascicle axis determine the length of these bristles. Here we can see the bristles. This is the hair-like structure is the bristles. This is outer one and an inner one, and there are two spikelets. So this whole structure is called as fascicles, and there are number of fascicles are present over the rachis, and this consists of the total spike or earhead of the bajra. So the anthesis of in this bajra is commences from the 1/3 of the apex of the spike and proceed towards the both way, and stigma emerges first and the anthesis is over within the 2 to 3 days. As we can see that stigma emerges first, that means the gynoecium mature first, leading to the cross pollination. This is followed by the first male phase in which the anthers from the perfect flower emerged out. As in bajra we can see there are two types of flower: perfect flower that having the male and female flower, and another one is having only male flower. So there are two flowers present, or two florets are present in the spikelet of the bajra, and the stigma mature first and they emerge first, and then after the phases of the anthesis, that is anthesis is always done in the different phases, in which in first phase the male floret from the male phase or the male anther from the perfect flower is emerged out, and on the 5th day of the anthesis the second male phase begins, in which anthers from the staminate flower emerge, and this anthesis time is the 8 p.m. to the 2 a.m. So this is the anthesis time. Anthesis is the first opening of the flower. So this anthesis is always happen in the phases. In first phase the perfect flower the male parent from the perfect flower are in the anthesis, and the second phase is from the staminate flower. You can see here the fascicles. These fascicles having the two florets, one is sessile and one is pedicellate, and these two having the outer glume, then inner lemma and pelia, and then 3 stamens and the bifid feathery stigma, having the stigma style and ovary. So this is the all structure, flower structure of the pearl millet, but it is very small. At the same time, in the pearl millet, there are two types of florets are present. Some of them are perfect one, and some of them are staminate or male floret. In perfect floret, the gynoecium and androecium present along with, and the gynoecium, that is the female part, is mature first, we can see here, and then after the anthers emerge out. The stigma is emerged out 2 to 3 days before this gynoecium, we can see here. But in case of the staminate flower, there are only the anthers, and they are emerged after this the perfect flower anthers emerged out. So this is the floral structure of the bajra or pearl millet. We can see here in diagram also there are stigma, the bifeathery or bifid stigma, then the staminate floret are here. We can see in the single spikelet, in that the anthers are present at the center. Here is the close up of this bisexual flower, bisexual floret having the outer glume and inner glume, and between these there are the anther, and at the base there are stigma. And we can discuss this about the fascicle earlier. We are earlier discussed about this. Then what are the crossing technique or hybridization technique which is used by breeder in the hybridization or hybrid seed production of the bajra? So we, up to this, we have already discussed that bajra is protogyny. That means having the maturity of gynoecium or stigma before the anther. So we can see here all the stigma are emerged out, and there is no anther presence. Means after the gynoecium, this gynoecium are matured, and then after the androecium is, that is anther, is emerged out. So there is as such no needs of emasculation in bajra. By using this trick, we can handle or we can produce the hybrid seed without emasculation. So emasculation is generally done to avoid the effect of the pollen in the bisexual flower, or the pollen contaminate the female. But due to the different in their maturity timing, the gynoeciums are mature first, and anthers are in the bisexual flower, is perfect flower, the anthers are not mature enough. So we can easily pollinate this stigma by using the foreign pollen or the another male parent, which is we can use in the hybridization. So these are the all female parents, and their anthers are still not mature, so we can use the another male parent and produce hybrid over here. So this is the case in case of the bajra, in which we need not have to do emasculation as such. So by using this protogyny condition, we can easily produce the hybrid without emasculation. The emasculation in pearl millet is laborious and difficult due to the small size of flower and the late maturity of the anthers when compared to the sigma. And about 4/5 of the upper portion of the spike is removed, and the rest is bagged before the styles are appear to prevent the contamination. So for that, producing the hybrid, there is a technique that suppose this is the earhead or spike of the bajra, then about 4/5 of the upper portion, that is 4/5 of the upper portion is removed, and the rest is bagged, that is 4/5 is removed and the remaining one is bagged, so that only females are produced or only females are available in the particular female plant. And by bagging them, we can prevent the contamination of other pollen, and the flowers are pollinated. So these flowers or these females are pollinated by dusting them with the fresh pollen obtained from the desired male parent. And when we want to cross-pollinate these with the desired male, we can dust the pollen from that particular male parent on the female parent. So by the crossing between these two distant parent, the F1 is produced on the female line, and or by shaking the spike which shedding the pollen over the exposed stigmas of the emasculated, sorry, not emasculated, here the only stigmas are present, and the over that particular stigma we can use the desired male to pollinate them or hybridize them and produce the hybrid seed. So this is the overall technique of the crossing, which is very simple, but before we doing the hybridization, we must to know all the flower structure and timing of the anthesis and stigma receptivity in the pearl millet for the successful hybridization. So thanks friends, this is for educational purpose only and references are as below. Thanks once again.